So what we've done is we've investigated some of the really important molecules that are inside cells. Where we go with area of study 2 is more into looking at the biochemistry. And by the biochemistry, we mean the chemical reactions that happen inside cells for cells to function. And of course, we know that nucleic acids, DNA code for proteins, and particularly the role of proteins in this story. And the role of proteins is all about enzymes. So that's what we look at first of all when we look at this sort of idea. We are particularly looking at, let me just change my color. We're particularly looking at the role of enzymes in this cell reaction because they are proteins and you know everything about how they're made, you know about their shape and all that sort of stuff. Then we look at some particular cell reactions. And the two cell reactions we look at are particularly photosynthesis and cellular respiration. These are the two key energy reactions, if you like. And so to understand the importance of photosynthesis in trapping energy into the ecosystem and how the actual chemical reaction, I hope you all have heard the word photosynthesis in your junior science, and you probably think of plants. We go into the cell and actually learn more about how the actual reaction happens. And then with cellular respiration, again, it's not just about breathing in oxygen and breathing out carbon dioxide. It's about the cell reaction whereby food, call it glucose, is broken apart to release energy for cell function. So we look at photosynthesis and cellular respiration as energy reactions, chemical reactions. Don't get concerned if you're not doing chemistry. The level of chemistry we need is very, very simple. However, if you are doing chemistry, you will also do these reactions in much greater detail. And that's fine. And it doesn't matter if you use that detail in biology answers. You don't get any extra marks because it's more detailed, but it's quite acceptable. You'll see that as your teachers go through it. So we move away from the DNA protein story and genes, and now we go into proteins for enzymes to drive reactions. What reactions? Photosynthesis and cellular respiration. So it's quite a different step into the world of Area Study 2, but it is linked by the knowledge of proteins. And that's the part that links both of them together, is understanding proteins and their importance in the world of cell function. So what I want to do in the last half hour, I particularly want to talk about the enzyme story, because this is the protein story. We have the notes for cell respiration and photosynthesis, and I think they're a really nice set of summary notes for you as your teacher goes through those. But I just want to touch again, as I say, onto the enzyme story, because that relates more to what we've done in our area of study 1. So I'm looking on what page am I on? Page 122. And just while you're turning that, I'll see if there are any questions that have come up in the meantime. Oh, good. Lovely. Okay. So, as I said to you, cells react in reactions called biochemical pathways. Now, you've probably done a little bit of chemistry, and you usually know reactants are the starting molecules in a reaction, and then we have an arrow, and then we write the products. And that's usually what a chemical reaction is written as an equation. How many reactants there are, it just depends on the reaction we're talking about. How many products, again, it depends on the reaction. But we usually use just a single arrow. We can do that with biology, except most of the reactions to go from initial reactions to the final products is not just one big jump. There's usually a series of steps. And in biological reactions, having a series of steps, what this does, it gives the cell greater control over the reaction rate. Because once again, a cell only wants the products in the quantities that it needs at a time. So sometimes it might need those products really quickly. Other times it may not need them much at all. Other times it may not need them at all. So this idea of just going from reactants to products is just one big jump. It happens or it doesn't. Having steps means that things can be controlled along the way. So what we have when we've got our idea of reactants and products, here's our first reactant. It will then become not a product, but what we call an intermediate molecule through a series of steps. And then that will become changed into another intermediate molecule through a series of steps, till eventually we come to our product. What controls each of these steps are these amazing molecules called enzymes. And every step has its own particular enzyme to run it. So this first step here, enzyme 1, whatever that is, will make that happen. The next step, enzyme 2, will make that happen. The next step, enzyme 3, will make that happen. If for some reason the gene mutation happened that enzyme 2 isn't being made, then the product can't happen. You must go through each step to get to the final product. So you can't jump steps. And as this picture shows, sometimes the product can also turn off the reaction. So when the cell's got enough of the product, just building up that product can feed back and turn off the reaction. So can you see how there's a lot of control to go from reactants to products if it's done in lots of steps? And as I said, each step is controlled by a separate, and there's the word, specific enzyme. And the reason the enzyme is specific is because it's going to match the shape of its substrate, that's reactant, molecule. So if you look at the diagram, the first reactant is a triangle, a green triangle. The enzyme has a shape to match it. The second is a purple rectangle. The enzyme matches that. The next one is a blue wobbly thing. The enzyme matches that. So what we've got to control these reactions are enzymes. And enzymes have unique specific shapes. That's why enzyme 1 can only do that first step. Enzyme 1 cannot do the second step. It doesn't match the shape of the substrate. Enzyme 3 can't do the first step. It doesn't match the shape of the substrate. So if we're talking about shapes, there's one molecule we know, it's all about shape. And that is because enzymes are proteins. So the first question often asked is, what is an enzyme? And the first statement you make is enzymes are proteins. But they're not just any protein. What they do is they speed up chemical reactions, but do not take part in the reaction. And going back to that question that was about the PCR, the DNA polymerase doesn't run out. It always can go back and redo because the enzymes do not actually become part of the products. They are always left at the end unchanged. So that's what an enzyme is. They are protein molecules that speed up reactions but don't take part in the reaction. That's a nice definition. Reactions will happen, but very, very slowly. Cells need reactions to happen at the correct rate, the optimum rate. We know when we're exercising, we need our reactions to be happening faster so we can have more energy to exercise. We know when we're sleeping, we don't need our reactions to happen quickly. We need them to happen much more slowly, so we're not wasting energy. All that, speed up or not, is what our enzymes are doing. It's the same reaction, but it's whether it's happening quickly or slowly. That's where the enzyme pathway is important. The second part of enzymes is this idea about enzymes being specific. And that's to do with the idea of each enzyme acts on one substrate or molecule, if you like, only. Enzyme 1 only acted on that green triangle. Enzyme 2 only on the purple rectangle. One molecule only. The reason is because of their shape. And an enzyme is a protein which was made as a primary structure. It then got folded into helices and alpha helices and beta pleated sheets, which was secondary structure, just like all proteins do. Then it went into the three-dimensional tertiary structure. And this is where enzymes finish. And each enzyme has a unique tertiary structure. And there's one part of the tertiary structure that is most important. And the important folding shape is called the active site. The active site is where the substrate joins to. And this has to happen for the step to occur. And so this unique tertiary shape of the active site is the key to the enzyme being specific. It matches only its particular substrate. So again, our green triangle substrate, the enzyme had the active site that matched, or complementary, same thing. The purple rectangle, the enzyme's active site matched that. And the blue one, the active site matched or was complementary. So this is what determines enzyme specificity, their shape. So it goes back to the part we were doing in error study 1, the shape of the active site. So enzymes are proteins that speed up reactions without taking part of them. They are highly specific. Each enzyme will act on one substrate only because the active site of the enzyme is complementary to that one substrate shape. So the question is, how do they do their job? Well, the enzyme attaches to its substrate. How they do it. Now, the main model or the main theory is called the lock and key model. And you know, if you've got a key, it only fits one particular lock. The lock is like the enzyme, the key is like the substrate. So the substrate will only fit into one particular enzyme's active site. And so here's how we like to do a flow chart. This is not a reaction equation. This is a flow chart of enzyme action. This is the lock and key model. So we start with the enzyme, and here's its active site. I have no idea why it's so complicated, but there you go. This is the active site, and I'm just writing AS to stand for active site. Here are our substrates. They're mixed together in the cell cytosol or wherever they might be, or even in a test tube. So they're mixing together. And what we have is the enzyme joins to the substrate. We call it the enzyme substrate complex. And you can see how those substrates have completely fitted into that active site perfectly. When that happens, what happens is that those substrate atoms move around and form products. So here's the product, excuse me, at the end, which is not the same as the original substrates. It's a different, complete thing. But notice the enzyme is unchanged. It can go back and do the reaction again to another set of substrates, and on and on. Students have been asked to draw this flowchart reaction in exams. If you do, it's an annotated diagram. Don't make it hard for yourself. Make a very simple enzyme shape with an enzyme and label the active site. Have a substrate, you don't need two, just have a substrate that has the shape that will fit the active site. So that's how I would do the first picture. Then you must show your enzyme with the substrate combined. Just do some labeling. See how I'm labeling things? That's what we mean by annotated. And we call this the substrate, enzyme substrate complex. And we say that the substrate fits the active site. So you write a little bit of annotation on there to explain. So it's not just a picture sitting there in the middle of the page. Then we come to our final picture where we have our enzyme and we write unchanged and can be reused. And now I'm going to put a product. And I make the product a different shape. So that's the typical lock and key model of how enzymes attach to their substrate, and then we get it unchanged and a product formed at the end. And if you're ever asked to explain it, the lock and key model is the one to explain. But there is another theory. And they think that most enzymes probably do lock and key, but there is another theory called the induced fit. Now, the reason there's this theory is because when they analyze cells, the number of enzymes in a cell does not match the number of pathway steps in the metabolic reactions. There are more steps than there are enzymes. And if we know steps are catalyzed by enzymes, you'd think for every step there would be an enzyme, but there's not enough enzymes. So what they're thinking is when a particular substrate comes in, the enzyme can actually change its active site shape to mold around the substrates. What this means is one enzyme can catalyze a series of steps. So it might mold to do the first step, then it might mold again to do another step later on. So this is a way to explain how can there be so many pathway steps? How can there be not enough enzymes if the enzyme molds to fit? But from this point on, it's the same. It complexes, and then we get products, and then the enzyme can be used again. So it's just this part here, the first step is a little bit different in that theory. But again, we do want you to do the lock and key at VCAA if you are asked to explain it. But it doesn't hurt to just have an idea of this other model that is possible. But none of that answers the question, how does an enzyme fitting with its substrate make a reaction go faster? Well, this is why on page 125, it's all about the activation energy. Now, to get from reactants to products, a certain amount of collisions is needed by the atoms. And when the atoms collide, the bonding inside those molecules actually break open and the atoms can rearrange. That's what a reaction is. You start with reactants, which have atoms joined together. They collide through kinetic energy, and then those atoms separate and we get new products forming, new combinations. That collision energy is called the activation energy. It's the energy needed for a reaction to happen. It's the amount of collisions, reactant collisions, if you like, that's needed for the products to form. What enzymes do is they reduce that energy. It means that the reactants don't have to collide as much before they become all separated and broken up. The reason why is when we have the enzyme and substrate joining together, when we have that complex, it actually weakens the bonds in the substrate. It makes them a bit weaker, which means they don't have to collide as much before they're broken. And so it makes the bonds easier to break and new bonds easier to form. So that's how enzymes speed up reactions, by lowering the activation energy needed. And here's a graph to show you what we mean. And this graph is one that you should be able to understand. These are the reactants, whatever they are in a reaction, and these are the products, like that. Now, for the reaction to happen without enzyme, we need lots of collisions and we need this much energy. Oh, I'll do it in blue. Oh, no, I'll do it in red. I'll match the thing. This is the amount of energy that's needed for the collisions to happen to then form products. That amount, whatever that is. When we add enzymes and it complexes, still collisions are needed, but not as hard and not as strong. It's just that amount of energy needed. And of course, that's going to get, the collisions are going to get there quicker than to the bigger part. Notice we still end up with the same products. We still start with the same reactants, but it's just how long or how much energy is needed to get the reaction to happen. So with enzymes, we have this much lower energy requirement to get the products forming. What is an enzyme? Enzyme specificity, and how do enzymes catalyze reaction? And just understanding the lock and key reaction as a way of enzymes doing their action. And that doesn't matter if it's DNA polymerase, DNA ligase, RNA polymerase, any enzyme this applies to that's happening in the cell systems. And it could be in the cytosol, it could be in the nucleus, could be anywhere around. All right, the last part is what can impact on the enzymes doing their job. And this sort of relates back to the PCR in some ways. There are 4 key factors that impact. The first one being temperature. And someone asked the question about the DNA polymerase, if it's not at 37, how can it work? And remember, we were using the TAC DNA polymerase in the PCR. That's because every enzyme has an optimum temperature. And this is species determined. All human enzymes work best at 37 degrees, which is what was said. I think it was Helen or someone said. If you're a bird, your enzymes work best at 44 degrees because that's the temperature. If you're a thermal aquatic bacterium, your enzymes work best at 90 degrees. So each species has an optimum temperature for all their enzymes. Doesn't matter if they're enzymes inside cells or enzymes in your digestive tract, all enzymes in humans work best at 37 degrees, which is why if our temperature goes up in a fever, we're not feeling too well because our cell reactions aren't happening properly. Or if we've got really cold, lower temperatures down to 34 or something, hypothermia, reactions aren't happening fast enough. With temperature, you are expected to understand what happens. And this is a typical temperature graph. So here we've got temperature increasing. This is a human enzyme graph. The reason I know it's human is because the optimum, which means the best or fastest temperature, is occurring at 37 degrees. So that's the optimum. Temperatures lower than optimum, this range here, as you increase the temperature, you increase the rate of reaction. As you decrease the temperature, you decrease the rate of reaction. So here we've got increased temperature. We're going to increase the rate. That's because there are more collisions happening between the molecules. And if there's more collisions, there's more enzyme substrate complexing. If you lower the temperature, you lower the rate because the molecules are moving slower and there's less complexing. So that's what happens with temperature up to optimum. When we go above optimum, when we move into this range, notice the reaction doesn't keep increasing. The reaction actually decreases. This is because above optimum, or what we call critical temperature, which is usually a few degrees higher, the enzymes denature. And enzyme denaturing means the hydrogen bonds in the protein shape. Remember, the secondary shape is hydrogen bonding. They will break. When they break, the shape of the active site alters because the shape is being held by bonding. So if you break the bonds, the shape will alter. And it is a permanent altering. And so it will no longer fit with the substrate. And because it's permanent, once an enzyme molecule is denatured, it's useless. And then more will get denatured as time goes on. And so what we have is not just a slowing down, but we have eventually a dropping of the reaction once all the enzymes denatured. And even if I went back to 37 degrees, it wouldn't start up again. Think of when you have an egg and you break the egg into a frying pan, and you've got the clear part, you've got the yellow yolk, but the clear part is protein called albumin. The clear part is there. When you turn the heat up, the clear part goes white. What you're doing is you're denaturing the protein, and it's going that opaque white color. If you turn the stove off and the frying pan goes cold, it doesn't go back to clear. It's permanently denatured. This is why we had to use that Taq DNA polymerase in the PCR, because when we took it up to 90 degrees, if we used human DNA polymerase, it would be denatured. And even though the temperature drops down, the denaturing has happened. This is why cell temperature is vital. And this is why we have mechanisms like sweating and all that. It's all about keeping your cells at 37 degrees. And it's because the enzymes are not working. And when you have a fever, it's too high. Just to finish off, some of the other things, pH. Now, pH, every enzyme in humans doesn't have the same pH. Optimum pH depends on location. In your stomach, the pH is 2, very acidic. So the enzymes work best there at 2. In your mouth, it's about 7.5. In your small intestine, it's about 8 or 9. Enzymes work in different locations at different pHs. In the cytosol, which are most of the metabolic enzymes, it's got a pH of about 7. It's fairly neutral. So in the cytosol, there has to be ways to keep the cytosol pH at that rather neutral stage. And there's lots of molecules dissolved there. So the cell has natural buffers to keep it at that pH. And then finally, the last two are the concentration of enzyme. The more enzyme you have, the more active sites, the faster the reaction can happen. But there will come a time where the enzyme's done all its work and there is no more substrate, so it can plateau. And the same with substrate concentration. The more substrate you have, the more molecules, the more collisions, the faster the reaction. But there will come a time when the active sites are all working at maximum. So it doesn't matter how much substrate you have, the enzyme can't work any faster than it's working. Factors affecting enzymes. And then, of course, we have things that help enzymes. And remember, we talked about vitamins and minerals as part of important to help our enzymes do their job, cofactors and coenzymes. And then, of course, just to finish, we have the nasty inhibitors, which stop enzymes working. These can be good because we can use these as painkillers to stop enzymes involved in the pathway of pain and that. But naturally, you don't want your cells to have inhibitors. You need your cell reactions happening as they should. Okay, it's time. Now, I just wanted to cover the enzymes, so because they relate back to the proteins for you. But as you go through Area Study 2, please come back into this book. Then we've got nice summaries about photosynthesis and cellular respiration, which hopefully will help you work through. Can I remind you that we're always here if you need help? And of course, we've got the masterclasses and all those sort of things that are happening through the year as well. So I will say good luck to you all. Let me just answer questions. Look, I think taking notes is fine. I think usually using ideas of pictures, making summaries, writing out keywords and their definitions, all that's good. Just writing out lots of notes is not very useful. Needing to make summary notes, that's what's important. Look, I don't know what makes the induced fit thing, how it molds around. I have never read as to why it molds and how it does that. So it's a great question. Minihill, but I don't know the answer to that. I'm sorry. I wish I did. Probably have a Nobel Prize if I knew that. Okay. Yes, the sessions have been recorded, and you'll be, I think there was an email sent out about the recording for you. Okay. If you have a look for questions, the responses are at the back. So hopefully the answers to that question, this is for Hannah, will be explained okay at the back. I need to let you go because it's now time to just chill and relax and enjoy your weekend, enjoy what's left of this beautiful day. And I wish you no, you don't need luck. Really enjoy your biology. It's a great course. It's a great year, 3-4 biology. Ask lots of questions of your teachers, offer lots of answers to those questions, and really just keep churning away. Don't leave things as you do a little topic, then get a little summary done of that. Do some questions from our book or VCAA or whatever, and just chip away as you go and working towards those SACs, and you'll be fine. So good luck to all of you. I've enjoyed my 2 sessions with you. Hope I've been of help. Bye, everyone.